Welcome back.
Okay,
All
I'm going to try again.
right. Excellent. Thank you.
Just bear with me while I get the PowerPoint back alive.
Hey, got to love this virtual world.
Oh, it's just so frustrating. Just
Had to have a bit
some
of
it
a crash
fails, and then you've got 85 people watching you. Right, let's try it again. My Mac is saying that my performance is reduced. I'm going to try this again. All right.
We can hear you well this time anyway.
Share screen. Let's try this again.
Excellent.
All right. Okay. So hopefully you can see my screen and hear me.
Yes, perfect. Thank you.
Okay. All right. So my name's Michael Dickinson. I'm talking about new treatments for aggressive B-cell lymphoma and also an update on CAR T. And just to give some background about me, I'm the disease group lead for aggressive lymphoma at Peter Mac, and also a member of the CAR T team, which started up there in the last year or so. I started at Peter Mac in 2007, and I've been there full-time since about 2010. And my main focus, apart from looking after patients day to day, is new trials of new drugs. And some of these trials I've designed and others are industry sponsored. And I've worked for Lymphoma Australia for several years, collaborating with Donna and Sharon around providing medical input to some of the work that Lymphoma Australia does, and also doing talks like these, which I've really enjoyed. So today I'll be talking about an overview of the space in aggressive lymphoma, talking about some of the promising new immunotherapies, which really is a theme here that you'll hear me talking about quite a bit. And I'd like to make the distinction early on between passive immunotherapies, of which rituximab is an example, and some of the very T-cell activating therapies, of which CAR T cells are an example. And through all of this talk, I'll provide a bit of a context around the Australian landscape. I think that Australia is pretty well represented in terms of having access to these drugs through clinical trials, and of course through the public healthcare system. But it's a field, particularly diffuse large B-cell lymphoma, which is the most common aggressive lymphoma, that has been a bit slow to develop new drugs over the last 20 years. Just in the last 18 months or so has developed, that we've seen some really important new developments for anti-cancer drugs in this space, which I'll be talking about. So why does it matter? Well, what is our challenge at the moment? When we're developing new drugs, we don't want to overtreat people with toxic therapy. We don't want to treat people if they can't tolerate intensive therapy. And we know that the higher our response rates early on, the better people go throughout the course of their cancer journey. That is, the first bite of the cherry, picking the right first treatment, is really the most important way to make a difference. And pleasingly, with our aggressive lymphomas, many or most of our patients are cured with frontline therapy, but of course, there's a significant proportion of patients who aren't cured. And the earlier we achieve cure, the better. So particularly with these aggressive lymphomas, what we're trying to do is offer patients a remission that is deep and durable. And our goal of treatment here is to achieve a complete remission, as opposed to some of the more indolent lymphomas, where the goal is a goal of preventing progression might be acceptable. In the aggressive lymphomas, really, we need patients to get into a complete remission. We also need to have better tools to understand which patients need more intensive therapies and which patients need which new therapies. So one of the challenges that we have is that diffuse large B-cell lymphoma, one of our most common lymphomas, is divided into at least 7 molecular groups that are determined by the fundamental genomic drivers of the disease. So what we think of as a single disease, diffuse large B-cell lymphoma, is probably at least 7 different diseases. And I think the future is probably going to mean tailoring our treatment to each one of these different diseases. And we're just not there yet in terms of being able to routinely pick apart the different types of lymphomas in our diagnostic process. And so it all comes back to these groups of lymphoma. And here I refer to a slide that I think I wrote years ago that I think is really important just to provide a framework. So I've been asked to talk about aggressive lymphoma, and these words, indolent, aggressive, and very high-grade lymphoma, really speak to the tempo of disease. And I'll come back to the tempo of disease, but in the aggressive lymphoma space, the space that I work in at Peter Mac, we treat diffuse large B-cell lymphoma, peripheral T-cell lymphomas, Hodgkin lymphoma, and mantle cell lymphoma for that group of patients who require more aggressive therapy. Mantle cell lymphoma is one of these ones that can behave like an indolent lymphoma and also as an aggressive lymphoma. So often when you attend a talk about indolent lymphomas or aggressive lymphomas, you'll hear about mantle cell lymphoma, which can behave in either way. But the most common lymphoma by far is diffuse large B-cell lymphoma, which accounts for roughly a third of all lymphoma diagnoses, presents at a median age of 65, has around a 65% cure rate with frontline therapy. And it's the one that we see most commonly in our clinics. So what are the problems for patients who relapse and for the doctors looking after them? Well, the first thing is that the disease kinetics are an issue. So this thing that I've been talking around, aggressive lymphomas, these are lymphomas that require treatment quickly before they cause a problem. And one of the challenges that we have in diffuse large B-cell lymphoma is that the disease tends to present with symptoms and then require treatment relatively quickly. And as a consequence, designing clinical trials that move fast enough to offer patients new treatments is very difficult. I can see in the Q&As that there's at least one person online who's been involved in a clinical trial, and they will know that it's typical for a clinical trial to want to ask the question, is this new drug safe, and does this new drug work? And and the way to do that, of course, is that you have to have a disease that can be measured, and you have to have time off other drugs so that you can tell that the new drug in the trial is the one that's working. And as a consequence, most clinical trials have something called a treatment-free washout window of about 4 weeks between the last chemotherapy and the new treatment on the clinical trial. The problem with that is, with some of these aggressive lymphomas, a subgroup of patients will have lymphoma that moves too fast so that they need treatment sooner than 4 weeks. And as a consequence, this has been a very difficult space to develop new drugs in, because it's been very hard to design trials with an adequate washout window from previous treatments that still includes patients with the most aggressive of lymphomas. And as a consequence, all of the trial data we have probably represents around half or 2/3 of all the diffuse large B-cell lymphomas that we see in relapse, but there'll still be a small group of patients with very fast-moving disease where it's very hard to get patients onto clinical trials. And in those situations, we really are turning to access programs for clinical drugs and of course, the pharmaceutical benefits scheme to make these drugs available to us so that we can immediately give them to patients when they need them. But it is one of the problems for getting people onto clinical trials. Another factor is that while we've become better at understanding the genomics of lymphomas, usually in a given person, there are multiple factors that drive the disease. And so we may be able to detect a particular genomic mutation and target that with a type of targeted drug, but most lymphomas have more than one way of getting around a novel treatment. So some of these very nice targeted agents like ibrutinib, which we see to be very active in chronic lymphocytic leukemia, are less active in diffuse large B-cell lymphomas because they have more mechanisms of action keeping the lymphoma cells alive. And that means we need to find treatments that kind of get around those single pathways of cell survival that we understand and actually target multiple things at once. Chemotherapy does that very well, but of course we know that some patients relapse after chemotherapy. Another real challenge in treating and developing treatments for aggressive lymphomas is patient fitness. I know that I'm speaking to a patient group, and you will know that the median age for some of these common lymphomas is 65, which means that half of patients are younger than that and the other half of patients are older than that. And as you get older in life, you develop more and more other medical problems, and they can get in the way of giving treatment because they render people more susceptible to treatment toxicities and less able to bounce back if there are treatment toxicities like infection or mouth mucositis or low blood counts. Those sort of toxicities become difficult to get around. So one of the challenges in clinical trials is trying to find treatments that are gentle enough, particularly in patients who've already developed some complications from prior chemotherapy. Right now, this is our current treatment paradigm for our most common lymphoma, diffuse large B-cell lymphoma. But essentially, patients who have the diagnosis are usually cured with frontline therapy. About 2/3 are cured with frontline therapy. For those who relapse, the standard treatment option is to assess whether patients are fit for autologous stem cell transplantation. It's a long series of words that basically means providing high doses of chemotherapy, but keeping the bone marrow stem cells to the side so that they don't receive the chemotherapy. This enables us to give anti-cancer chemotherapy that are several-fold higher than our standard doses of chemotherapy, and to do it safely. But because the toxicity of the chemotherapy is greater as we increase the dose, for younger patients, they find autologous stem cell transplantation bearable. But as people get older in life, autologous stem cell transplantation isn't the right treatment for everyone who relapses the first time. Now, we do transplant patients up to the age of 75 or so. We've transplanted patients older than that at Peter Mac, but it tends to become more difficult to do the further past 70 that someone gets. So one of our challenges here is what to do about patients who aren't fit for autologous stem cell transplantation. And if we consider that autologous transplantation cures roughly 40 to 50% of patients, it's what to do with patients who relapse after autologous stem cell transplantation. And essentially at the moment, and I've deliberately put up as the 2018 treatment paradigm, there is a group of patients for whom there aren't great treatment options. That's patients who aren't fit for autologous stem cell transplantation and patients who relapse after autologous stem cell transplantation. Now, an important part of autologous stem cell transplantation is that we need to be able to control the lymphoma before we move patients into that therapy. So if a patient has a relapse of diffuse large B-cell lymphoma, and this is also true for Hodgkin lymphoma, I hasten to add, then we use standard doses of chemotherapy to get the lymphoma under control before we move to the intensive chemotherapy. So the other area of need is patients who have disease that doesn't respond to standard doses of chemotherapy, because we don't take those patients to autologous stem cell transplantation. So I know there'll be people on the call who are in that group, which, you know, it's a challenging and uncertain time. And one of the fundamental problems is that chemotherapy at the moment just isn't good enough, with only a small fraction of patients really having good outcomes if we just use chemotherapy after they relapse after frontline therapy. And there have been no new drugs for diffuse large B-cell lymphoma until relatively recently. And even in mantle cell lymphoma, there's really only been a few new drugs available in the Australian context, of which ibrutinib is the best example. It's a non-chemotherapy treatment that we use for patients when they relapse. Now, when I'm talking through some of the updates that I wanted to give you, I use this slide a lot, and I like to come back to it, particularly when talking to an audience that includes lay people. That is because it's really tempting to hear about new treatments and assume that they work in everyone, or really tempting to hear about treatments that appear to work in a lot of people, but not realizing that the clinical trials are either still underway or are very small and have very poor value. Now, we're talking here, I've been asked to talk about aggressive lymphomas, and there's lots and lots of different types of aggressive lymphomas. But for diffuse large B-cell lymphoma being one of the most common ones that we get, we're trying to get clinical trials that are randomized, double-blind, controlled trials. But in reality, the sort of trials that we get are randomized controlled trials and sometimes single-arm controlled trials. And the important thing about understanding the kind of trials that are happening in this space is understanding the challenge that doctors and funders have in determining how good the drugs are. If you give 10 patients that you choose a particular drug and they have a high response rate, that might be partly because you chose 10 good patients. If you chose 1,000 patients, then it's more likely that you'll have a mix of all different types of patients in that group. And if you randomize 500 patients to receive one treatment and 500 patients to receive another treatment, that's the best level of evidence. And surprisingly, we're seeing in one of our most common lymphomas that drugs are being approved on the basis of fairly small single-arm trials rather than randomized control trials, or they're being approved on the basis of very, very small randomized control trials. And this makes it really difficult for public funders like the Pharmaceutical Benefit Scheme to assess these treatments and assess essentially their value for money. And they are, of course, all ridiculously expensive at the moment. So this is a video, and in the video you can see green T cells killing tumor cells which are clear, and those tumor cells which are clear turn red when they begin to die. That's a reporter probe that shows when cells are dying, and what you can see is that the green T cells are going around killing the tumor cells and then going on to the next tumor cell and killing that, and then going on to the next tumor cell and killing that. And this brings me to a real theme in some of the new therapies that we've seen, which is that T cell killing is a really important way to target a number of different types of lymphoma in a way that may be relatively agnostic to what it is fundamentally that drove that lymphoma to exist. So if you take a drug like ibrutinib, which targets the B-cell receptor in chronic lymphocytic leukemia and in mantle cell lymphoma, and give it in diffuse large B-cell lymphoma, it's only active in a very small proportion of patients. And that's because the B-cell lymphomas develop a mechanism of resistance around that particular targeting point. What I think we're seeing with the immunotherapies is that they're able to kill lymphoma cells of all different types, which means that the immune system may be a new way to target these lymphomas in a durable way. So CAR T cells is one of the topics that I'm talking about today, and CAR T stands for chimeric antigen receptor T cells. And here the idea is that we have a designer receptor that has been developed specifically to target the lymphoma cells and to get those T cells to go and kill the lymphoma cells. And in this particular situation, the designer receptor is a chimera between the T cell receptor, which turns on the T cells and makes them kill their target, and an antibody just like the antibodies that we think of in vaccines. And essentially we've, through genetic modification, created a chimeric receptor, which is a hybrid of both receptors. And that designer molecule can be inserted into your T cells to turn them into CAR T cells. And in the case of the first approved product, the target for CAR T cells is this target called CD19. And CD19 is a target which is a protein that is expressed on the surface of B cells all the way from when they're pre-B cells, which is what we get acute leukemias from, all the way up to mature B cells, which is what we get myeloma from. And this bit here, these native B cells, activated B cells, GC-like B cells, are the type of cells that lead to our common lymphomas: diffuse large B-cell lymphoma, mantle cell lymphoma, chronic lymphocytic leukemia, follicular lymphoma, marginal zone lymphoma, and Waldenstrom's macroglobulinemia. And all of those lymphomas have CD19 on the surface of their cells, but also normal B cells have CD19 on the surface of those cells. And the first CAR T cells treatments that we know have been approved, and I'm assuming a little bit of background knowledge amongst the audience, targets this CD19 protein. So how does it work? Well, CAR T therapy begins with a blood collection. We take, using either peripheral blood or apheresis blood product, we take the cells from a patient using apheresis, freeze them down, and send them off to manufacturing, where the T cells are isolated. These T cells are the arm of the immune system that kills cells that you saw. They're isolated and infected with a virus that makes them express the protein on the surface of their cells, and then they're basically grown up in the lab, a little bit like the sourdough that my wife's been getting stuck into recently. And so they grow in the lab until there's a decent dose, and then they're checked for quality assurance and then sent back to the patient, back to the home hospital. At the moment, it's laboratories in America that are manufacturing the commercial products. The cells come back to the treatment center. The patient receives a little bit of gentle chemotherapy to remove their normal T cells, and then we infuse these CAR T cells as a single infusion of a product that's between 10 to 50 mils in size. And there are two CAR T products that have been tested in diffuse large B-cell lymphoma. One is tisagenlecleucel, which is also called Kymriah and made by a company called Novartis. In this clinical trial of roughly 130 patients, around 100 patients got the treatment, and essentially the pathway for a patient was as presented. Patients screened in the clinical trial and had their apheresis T cell collection done, and then were enrolled in the clinical trial and given chemotherapy to hold their lymphoma from growing while the CAR Ts were being manufactured. And then the CAR Ts were manufactured, patients received this lymphodepletion low-dose chemotherapy, and then the T cells were infused, and then patients were followed up with follow-up scans. Now, what's really important to understand, and it goes back to the first point that I made, is that the kinetics of the lymphoma determine the ability to give the infusion at this time. So CAR Ts can be manufactured in 2 to 3 weeks. So what you have to do is control the lymphoma with a bit of chemotherapy for that period of time. What we're finding in Australia at the moment is that it can be several months for these CAR Ts to be manufactured, which means that a patient needs to have the kind of lymphoma that is moving slowly enough that we can use standard chemotherapy to hold it while we're waiting for the CAR Ts to be manufactured. And that's been a real challenge day to day in providing this therapy, which I'll come back to. The other clinical trial that that currently led to a product which has recently had TGA approval in Australia is this trial called Zuma-1. Sorry, bear with me. My computer is having a fit. There we go. Zuma 1. And Zuma 1 uses a CAR T which also targets B cells. This particular one is made by a company called Kite, which is a company that is part of another company called Gilead. And in this particular clinical trial, patients were not allowed to receive chemotherapy to hold their lymphoma. So it possibly, arguably, recruited patients with lymphoma that wasn't moving as fast, but much more reliably in this clinical trial, the company was able to deliver CAR Ts within 3 or 4 weeks of beginning manufacturing. And again, the treatment population was much the same. We've got a question which was, do patients have to have a treatment-free window before leukapheresis? And the answer to that is yes, they do. So if one of the issues is that at this moment of leukapheresis, which is where we collect the healthy lymphocytes to turn them into CAR Ts, we don't want anything in the patient that affects the health of those T cells to undergo modification and growing in the lab. And so if a patient has no T cells in their bloodstream, that's not the right time to collect them. They need some recovery time. And it's normal for chemotherapy itself to lower the T cells. Also, radiotherapy lowers T cells. So you can have a quantitative low level of T cells, or you can have qualitative T cells dysfunction, which affects their ability to be manufactured. And the best example of that is probably a drug called bendamustine, which can affect the health of T cells. And we think that patients who've had multiple lines of treatment are more likely to have cells that don't grow up in the lab. Anyway, Kymriah has been approved in Australia and is funded and is available at Peter Mac under a government funding scheme. And Yescarta has, which is the drug made by Gilead called Yescarta, or axicabtagene cilolacel, has TGA approval, and we're waiting to see how that goes in terms of funding. Both of these treatments have common side effects which arise directly from the immune system being switched on. One is something called cytokine release syndrome, which presents a lot like infection and begins with a fever and may become serious and require intensive care, but doesn't require intensive care in the majority of patients who've received the standard commercial drugs. And another is neurotoxicity. So this is a slide that I've taken from a colleague, which shows that a patient with normal handwriting can lose their handwriting as an early sign of motor and cognitive dysfunction that arises because of the immune system being switched on. Usually this neurotoxicity arises in patients with cytokine release syndrome, and so usually a patient has a fever before they develop this neurotoxicity. But this can be very serious and life-threatening in a small but significant minority of patients. And what we have found is that low-level cytokine release syndrome is like having a really bad flu. And so if you have a patient who's already deconditioned and underweight, I've seen patients lose 10 or 15 kilos of weight if they've had sustained cytokine release syndrome or serious neurotoxicity. And so that's one of the reasons that this is not a treatment for all patients. This is a treatment where the patients who are fit enough to have it have to have good kidney function, good liver function, be on their feet and functioning well, so that if they develop one of these often unpredictable side effects, they're able to tolerate the treatment. Now, these drugs were approved on the basis of single-arm, non-randomized clinical trials of roughly 100 patients. And essentially they were approved because they increased the number of patients who were doing well above what could be expected with conventional chemotherapy. So just using chemotherapy third or fourth line and not it doesn't lead to good outcomes in the majority of patients. CAR-Ts are more effective, but still not effective in the majority of patients, to everyone's disappointment. So this gives us a real step forward in terms of outcomes for patients with relapsed diffuse large B-cell lymphoma, but not good enough for me and for many of my patients, something that we still need to work on. Nevertheless, the paradigm for treatment of diffuse large B-cell lymphoma has changed since these became routinely available in Australia, and they've certainly changed in the United States and Europe, where it's more widely available, such that if patients are unfit for autologous stem cell transplantation, they may be fit for CAR-Ts and get a treatment from that, or if they relapse after autologous stem cell transplantation, they can move to CAR-Ts and have a potential road to cure through that mechanism. We've certainly seen that CAR-Ts can provide durable remissions for a significant subset of patients for whom conventional chemotherapy might not have been effective. So the challenge for CAR-Ts is that they're complex to deliver. They have complex logistics. Right now in Australia, there's only one centre that's offering them for lymphoma outside of the clinical trial context. I work with a big team of physicians, a neurologist, we have a nurse specialist, and the intensive care are very involved in our routine care of patients. This is not something that every hospital can deliver, and it's also very expensive. So the calculated costs of CAR-Ts in the United States have been somewhere between 800,000 to $1 million Australian dollars per patient. We don't have perfect knowledge of what they cost in Australia, but they're likely to be very expensive for some of the more complicated patient journeys. Nevertheless, if CAR-T can do better than chemotherapy in the setting of third or fourth line treatment, maybe it can do better than autologous stem cell transplantation in patients who've had their first relapse. And one clinical trial that's open in Perth, in Sydney and in Melbourne is this trial called Belinda, which is a randomized clinical trial of autologous stem cell transplantation versus CAR-T in patients with their first relapse of disease who are fit for autologous stem cell transplantation. Here, the randomization is 1-to-1. There's 318 patients being randomized globally. There's dozens and dozens of hospitals globally that are running this clinical trial, and it is underway. It's been suspended for a short period of time, we hope, because of the coronavirus thing, and hopefully it'll be kicking off again and allowing us to recruit patients to it. But this is a standard option for my patients who relapse within the first 12 months of their original diagnosis, because we know they're the patients most in need of something better than autologous stem cell transplantation. How are CAR-Ts changing? Well, early this year, there was a report in the New England Journal of Medicine of this new treatment called CAR-NK cells. Natural killer cells are another cell in the immune system that can kill cancer cells. And these CAR-NK cells are transduced with this CAR receptor, but they're NK cells and they work differently and they use different hormones to divide and kill the cancer cells. And in the 11 patients, the small number of patients presented in this clinical trials, a significant number responded without any of the toxicities of CAR-T cells. And so while these treatments were custom made for the patients, which means that the patients have to have disease that can hang around waiting for treatment, this is a particularly exciting development in the cellular therapy space that we may see translate in the future in Australia. And there's a number of researchers in Australia that are trying to develop new CAR-NK cells. Right now at Peter Mac, our approach to the integration of CAR-T cells into the therapy of patients with diffuse large B-cell lymphoma is that if we have a patient with high-risk diffuse large B-cell lymphoma who we don't think will fit into that 65% who are cured with frontline chemotherapy, we offer CAR-T cells in a clinical trial in patients who have a positive PET scan after 2 cycles of CHOP or EPOCH. in patients who have less than a partial response after 4 cycles of CHOP or EPOC in that Belinda randomized clinical trial, or in patients who relapse within 12 months of their first treatment when they go into that Belinda clinical trial, or in patients who relapse after autologous stem cell transplantation when they may have an option for the government-funded CAR-T or other clinical trials we have of CAR-Ts at the moment. We hope that more hospitals will come online to deliver CAR-T therapy to patients in the immediate short term. But even if they do, we'll still be left with this problem of the patients who have lymphoma that moves too fast for the manufacturing of CAR-Ts, or where we have trouble collecting lymphocytes, as one questioner referred to. Here, what we've learned from CAR-Ts is that the immune system can kill cancers and can be harnessed. And in the last couple of years, we've seen an explosion in a new class of drugs called bispecific agents. And this all began with a drug called blinatumomab, which really showed that you could put patients with acute lymphoblastic leukemia into a remission by targeting that CD19 on the surface of B cells by using a small molecule drug that would bind the B cells to the T cells. And that drug works very well in ALL, but not sufficiently in diffuse large B-cell lymphoma to achieve the same sort of effects that we've seen as CAR T cells. But over the last couple of years, we've seen a series of follow-on drugs, of which this drug is an example, present the initial data at international conferences. So this is a drug made by the company Roche, it's called mosunetuzumab, and it's been in clinical trials at many centres around Australia. This is an antibody which binds T cells to B cells and turns on the T cells and makes them kill the B cell cancers. This particular drug has been tested in a complex clinical trial, and the key thing that I wanted to show you was that in aggressive lymphomas at a range of doses, it's actually much more active than we would expect from standard chemotherapy. It's given at low milligram doses and still able to induce complete remissions in around 20% of cases. And importantly, those complete remissions are sustained for a long period of time. And we've seen activity in diffuse large B-cell lymphoma, transformed follicular lymphoma, mantle cell lymphoma, and in Richter's syndrome. And this particular class of agents is very promising, and there are a number of companies that are making similar drugs. It looks like even patients who've failed, where CAR-Ts have failed, this drug is active in inducing remissions, and vice versa. There are cases where patients, where this drug has not worked, where mosunetuzumab has not worked, where CAR-Ts have worked. And as a class of agents, there seems to be more activity in patients with indolent lymphomas like follicular lymphoma than there is in aggressive lymphomas. But there are, as I've said, multiple of these types of agents being tested in clinical trials, and we are seeing a difference begin to emerge based on the chemistry. Now, one trial that I was involved in, and which is still running, is a trial of a drug that's just been given a name called glofitamab. It's actually made by the same company, Roche. This is some data that I presented last year that's going to be updated in the next couple of weeks at the European Haematology meeting. This antibody is a bit different because it binds 2 CD20s to 1 CD3. And that's kind of interesting on a molecular level, but from an outcomes point of view, what was interesting about this particular clinical trial is that it recruited mostly diffuse large B-cell lymphoma and transformed follicular lymphoma, and was able to induce a remission rate that was similar to what we see with CAR-Ts, so roughly 40%. And most patients at initial snapshot of follow-up have retained that complete remission. You'll notice these are much smaller numbers of patients. We now have many more patients worth of data that we're hoping to present in the coming couple of weeks, which shows, I think, that this off-the-shelf drug that you can give the day after you see a patient rather than 6 weeks to 3 months after you see a patient, may provide responses and durable responses that may be comparable to CAR T and more feasible than CAR T, because it doesn't require all of the baloney that it takes to set up a cellular therapy service. Now, these treatments do cause cytokine release syndrome which has to be managed very carefully and can cause some neurological toxicity, although the neurological toxicity is far less common than what we see in CAR Ts. Before I move on from these T-cell activating agents, there was a question here which said, can CAR T be used in patients who've had both autologous and allogeneic stem cell transplantation? The answer is yes, it can. It certainly can outside of clinical trials. Within clinical trials, they often exclude patients with allogeneic stem cell transplantation because of the toxicities that are left over from the allogeneic stem cell transplant. Patients who have graft versus host disease from allogeneic stem cell transplantation who require prednisolone or other T-cell modifiers to suppress graft versus host disease, those drugs may kill the CAR T. So long as a patient's not requiring those drugs, then CAR T can be delivered after allogeneic stem cell transplantation. Someone's ringing me. I'll just make sure it's not Donna. Like, it's not. Okay, so and I wanted to talk now about another drug called polatuzumab vedotin. And this is the idea, it's again built on the idea that you can target B cells on the surface using particular proteins on the surface. And now instead of CD20 or CD19, we're targeting CD79B. And this time polatuzumab vedotin is bound to a toxic agent, which basically is extraordinarily toxic on the cells. So it's given at micro doses and targeted on the surface of lymphoma cells and delivers this toxic payload to the cells. And what we see here is that you can essentially deliver chemotherapy at a much higher cellular dose level and not deliver that chemotherapy to other normal cells. By delivering this targeted payload, you can probably kill lymphoma cells in patients who've previously been refractory to chemotherapy. And I can see that I'm running a little bit over time, and I'm not going to go into too much detail about this drug, except to say that it is approved in the US and is currently under review in Australia. And the evidence for its approval basis is a very small randomized clinical trial of 80 patients, 40 in each arm, that look to combine this new drug, polatuzumab, together with bendamustine and rituximab, versus bendamustine and rituximab without polatuzumab. Now, bendamustine and rituximab is a relatively well-tolerated treatment that we use in B-cell lymphomas, more in follicular lymphoma than diffuse large B-cell lymphoma, but it is active in a small proportion of patients with diffuse large B-cell lymphoma. And what this trial showed, importantly, was a significantly higher response in patients who had received the new drug polatuzumab vedotin in combination with bendamustine and rituximab. And for a significant proportion of patients who had that complete remission, as you can see there, it was more than doubled the number of patients who got a complete remission. That remission is maintained. So I think a very important treatment option for patients where CAR T cells have let them down, or who are not fit enough to get to CAR T cells because of other organ problems. This could be a very effective treatment. I've used it a bit and have found it to be fairly well tolerated and a drug that is fairly easy for clinicians to understand and deliver. Another drug that has been reported at international conferences and in papers recently and has been in clinical trials in Australia is a drug called tafasitamab, which is also called MOR208. And this company, MorphoSys, have developed clinical trials to assess this drug as monotherapy and in combination with other drugs. And as monotherapy, it's not that good, but in combination with other drugs, it's significantly better. And while the trial in Australia combines it with bendamustine, that same chemotherapy backbone that we saw with polatuzumab, in this particular data set, it was a combination with lenalidomide, which is a drug that's usually used in myeloma, is moderately active in lymphoma, but not good enough to come to market in lymphoma. And in this combination, the idea is that lenalidomide turns up the immune system and the activity of the antibody. And impressively, really, I think it showed a complete remission rate, not that different from polatuzumab and bendamustine, around 40%, and a very significant proportion of patients were able to attain a durable response. So here we've gone from 2016, no great treatments for patients who relapsed after autologous stem cell transplantation, to CAR-Ts, multiple drug companies bringing out these bispecific antibodies that turn on the immune system as an off-the-shelf way of killing lymphoma. Now we've got polatuzumab vedotin, which has a targeted payload to lymphoma cells, clearly able to induce remissions in patients who are refractory to chemotherapy in a proportion of patients. And now we've got this other drug, or at least this drug combo of lenalidomide together with MOR208 or tafasitamab, which also seems very promising. And we don't see this routinely available in Australia, but I think we'll see more and more about this particular treatment. So I'm just going to pause there for a minute and just make sure that there's no things flashing on my screen, which there aren't. I'm going to switch directions now and just talk very briefly about mantle cell lymphoma. Mantle cell lymphoma is one of the rarer lymphomas that occurs again later in life and can present either as an indolent disease that may not require treatment, or as a more aggressive lymphoma that can be quite difficult to get into a remission and cure. And I just wanted to touch on a clinical trial that was presented at ASH last year, the American Society of Haematology meeting last year, which was really very striking. Right now, for patients who have relapsed mantle cell lymphoma, the treatment option is standard chemotherapy. There's a drug called ibrutinib, which is a good targeted oral agent that can induce a significant percentage of remissions in patients who are refractory to chemotherapy or have relapsed after standard therapies. This was the first clinical trial to test CAR-T cells. And basically, what's been happening with mantle cell lymphoma is different scientists have been trying different treatments, drugs like venetoclax and ibrutinib, to try to improve upon the outcomes of ibrutinib alone, and getting good, durable, complete remission rates, but still a proportion of patients relapsing ultimately after those treatments. This clinical trial took a CAR-T cell that's a bit similar to Yescarta, but not quite the same as Yescarta, called CTX19. And the striking outcome of this clinical trial was, again, the response rate. So an overall response rate of 93% after CAR-Ts in 60 evaluable patients, 2/3 of whom had a complete remission, and with complete remissions shown to be very, very durable in the ZUMA-2 population. So for a younger population, this is an important potential treatment option for mantle cell lymphoma, which I hope will have available soon for Australian patients with mantle cell lymphoma. Just wanted to touch briefly on clinical trials that are currently open. We have our own mantle cell lymphoma CAR-T trial that's just opened at Peter Mac. We've recruited our first patient that's of ibrutinib with CAR-T cells using the Kymriah product. We have something called allogeneic CAR-Ts, which uses unrelated donors and gets around that issue of manufacturing time, by training other people's CAR Ts to give to the patient so that we don't have to go through that apheresis process for a particular patient. That's in phase 1, first in human trials, and we recruit sort of a patient every couple of months to a trial like that. So these are not widely available trials, but they are active. We've got follow-on products from the Novartis CAR T, and we're obviously looking at CAR T treatments outside of lymphomas as well. In the diffuse large B-cell lymphoma space, we have a trial of glofitamab, one of those bispecific antibodies, where we're using it alone or in combination with a drug called Gazyva, or with another bispecific antibody that turns on the immune system, or in combination with chemotherapy. They're probably the most interesting clinical trials at the moment. And the other really important way of harnessing the immune system that we're just learning about is macrophage checkpoints, which a bit similar to drugs like Keytruda, can turn on this particular arm of the immune system, which seem to lead to responses in a significant proportion of patients. There's a number of macrophage checkpoint inhibitors in clinical trials at the moment around Australia, and I think that will be another way of turning on the immune system. I think I'm right on 50 minutes, and that's not including IT issues. I need to acknowledge that I work within a large department at Peter Mac with lots of people who support me doing the things that I do. It's not something I could do alone. In the CAR T service, there's lots of doctors and technicians and scientists, and also in the clinical trials unit, I'm backed up by a large team of nurses who do all of the work really in helping patients onto clinical trials, who I'd like to acknowledge today. And I think I'll stop there and see if there are any questions that I can help with.
Thank you, Michael. It was fantastic, a great overview, and there's a few things I've learnt as well along the way too. So I didn't realise a couple of those trials were open, so that's good. We do have a couple of questions that came in before the day. Most of them
Yes.
are answered, so that's been great. One of them is a couple of people have asked, what is the success rate of CAR T cell therapy with diffuse large B cell or any other aggressive lymphomas, really?
We think that the success rate probably is linked to how early in treatment you have it. So if you have, if we take all the patients who are in the pivotal clinical trials, there did seem to be a connection between patients who'd had 5 prior lines of therapy not having as high a success rate as patients who'd had 3 prior lines of therapy. So the first thing to say is there are factors that we've identified that affect that success rate. Having less lymphoma is better than having more. Having no symptoms is better than having some symptoms. Having the kind of lymphoma that didn't require chemotherapy while the CAR Ts were being manufactured is better than having a lymphoma that required lots and lots of chemotherapy while the CAR Ts were being manufactured. But in all comers in the clinical trials, the success is not just responding, it's keeping the response. And so if we look at the 12-month complete remission rate, it's somewhere between 1/3 and 40%, depending on the products you look at.
Okay.
And much higher, as you can see, in mantle cell lymphoma, which is interesting.
Yeah, it's looking very good results for mantle cell lymphoma. Also, another question has come through as well. With COVID-19, has this affected the amount of patients are able to get CAR T because of the flight restrictions and all that sort of thing, or how's it look?
With COVID-19 getting access to which treatment?
After CAR T cell therapy.
So it's complicated. So it's definitely complicated. So as you can imagine, the answer to that question seems to have changed every week. That's the first thing to say, just like everyone's lives. Secondly, the stakeholders in the answer to that question are state government and hospital based. As a state hospital, we need to follow the general guidance of the states. So in short, we try to avoid interstate travel unless it's necessary. we've taken a view at Peter Mac that while we're the only centre that's open, we try to treat interstate patients as if they're locals as best we possibly can. And COVID-19 hasn't changed that attitude. We have tried where possible to prevent the number of trips across borders by using telehealth as the first consult, particularly where it's unclear that everyone's on the same page in terms of how things are likely to work. I would say that more than half of our patients at the moment are interstate patients in spite of COVID-19.
Okay, thank you. And
there's a lot of positive will within the hospital to provide a national service.
it's fantastic, and especially with patients being spread out everywhere at the moment. It's a lot of uncertainty, that's for sure. And also, is there an opportunity maybe in the future for CAR T cell therapy in T-cell lymphomas?
Yeah, so this is difficult. One of the problems is that if you look at the paradigm in B-cell lymphoma, we're taking a target which is present on normal B cells and on the cancerous B cells. And the CAR Ts kill both. It just so happens you don't really need your normal B cells that much. You can live without them. There are complications, which I didn't go into, but you can live without them. With T cells, the issue is fratricide. That is the idea that the CAR Ts kill themselves because they bear the target that they're going for. And so the challenge in T-cell lymphoma is to find a target that that is on the cancer cells that isn't on the CAR T cells. And there are a number of targets being explored, and certainly we are trying to get targets from outside the country. We know of CAR T cell trials that are active in this space. One target is the target that brentuximab vedotin targets, which is CD30, which we know from that experience is a viable target. And we've seen that CAR T cells targeting CD30 are active, particularly in Hodgkin lymphoma, anaplastic large cell lymphoma. Unfortunately, T-cell lymphomas don't all express CD30 very much, and so there are other targets that are being explored.
Okay, good. Would you think that in future there might be some trials that be looking at bispecifics first, and if relapse from that going on to CAR T, would that be a viable?
Yeah, so look, I feel that intuitively people, these treatments might fail in patients where there's a problem with the target. So we know that lymphomas can change their spots, or might fail where there's a problem with the quality of the T-cell killing, which could be due to the lymphoma having a fundamental resistance. You know, we know that some lymphomas are fundamentally resistant to the immune system, or could be something fundamentally weak about a patient's T cells. So I still feel the question of sequentially treating with different immune treatments is an open question. What I would say is that the history of combining new drug with rituximab CHOP is a bit vexed. We actually do have a clinical trial exploring that particular combination at Peter Mac at the moment. It's in a safety phase, so it's only for patients with relapsed disease initially. But once that's finished, it will flip into a frontline clinical trial using glofitamab in combination with R-CHOP. I think that's a very exciting treatment option. What I hope is that increases the cure fraction from what our goal is as cancer doctors is that patients don't relapse at all. So what we really, really want to do is have a treatment like CAR Ts or glofitamab in combination with chemotherapy or mosunetuzumab or whatever bispecific, and actually cure such a great fraction with the frontline therapy that we don't need second line. We don't need to plan for second line therapy. I certainly don't think we should be pulling our punches with frontline therapy. We want to cure the majority of patients. So in short, yes, I do think these treatments are fit for moving to the front line. Absolutely, I do. And we're seeing that happen As I've mentioned, we've got a frontline CAR T trial at Peter Mac where we have our highest of high-risk patients have 2 cycles of chemotherapy, and if they're still positive on a PET scan, get CAR T straight away. So we are trying to move these treatments into the frontline. But when you've got chemotherapy curing 2/3, it's an uphill battle for any new treatment to beat that.
And financially as well, yeah.
Well, yeah.
Last question just came through, so this will be the last one. Has CAR T been used for double hit relapse less than 6 months?
Yes, so there's no routine availability of that treatment, but that Belinda trial recruits that patient group. So Zuma 12 is available. Zuma 12 is the frontline study. That's for double hit who are PET positive after 2 cycles of treatment. But if patients miss that trial because they're responding to treatment and then go on to relapse within the first 12 months, they're eligible for the randomized clinical trial of CAR T versus chemotherapy. And that includes double hit lymphoma. The issue that I'm finding with double hit lymphoma is that kinetics issue. There are some patients with double hit lymphoma who have a disease that moves at a pace that just beats the manufacturing pace of CAR Ts. And in those particular patients where I have the sense that that's going to happen, I do think that the signal from drugs like glofitamab and mosunetuzumab and their related drugs, I have to be careful here because I obviously have researched in one particular one, I do think the signal is strong enough from some of those other T-cell activating agents that I would feel comfortable reaching for a treatment that I can use tomorrow rather than a treatment I can use in 3 months. And sometimes when I've spoken to patients in this situation, your questioner has asked about, that's been my recommendation because a bird in the hand is the way to go. Donna, there's a question on the online Q&A thing. Do you want me to answer that question from Anonymous?
Go for it, yeah.
It says, how confident can we feel that our haematologists and their teams in major hospitals are informed about these new treatments and able to meaningfully consider them for various patients? How is all this information shared between research facilities and doctors on the ground in other states? This is a really difficult question. I think that it is a fundamental tenet of medicine that we all try to keep up to date and understand what's happening in our jurisdictions. I think all treating haematologists have a responsibility to do that and to do continuous learning. It is really hard for us to disseminate this information. It changes week to week. I think all patients should be, I wouldn't say lacking in confidence, but always have a healthy skepticism and always check in with their doctors about what they know about the current clinical trial seen across town and across the state and in other states around the country. There'll be doctors with a special interest in lymphoma who are totally up with it, and there'll be doctors who treat a broader range of things because of the treatment setting, where sometimes focus questions need to be answered. And I guess how confident can we be? The answer is, as a haematologist, I try to disseminate the information, but I'm not universally confident. I actually think it's a problem. I think Donna and Sharon have heard me talk about equity of access. And I think equity of knowledge is a huge issue, and a challenge, to be honest. And I think organisations like Lymphoma Australia help empower patients to get around that.
Yep, totally, and it's changing a lot too, and quite so quickly as well. It's very hard to, if you're not immersed in it, to keep up to date. So yeah, no, totally, and also coming to organisations like us will try and keep you in the know as much as we can. Thank you, Michael. That was a fantastic overview, and it's great to see some great stuff coming through in the pipeworks as well. It's very promising. A few comments from patients as this makes them feel a lot more assured that there's more coming. So I'll just finish off with this last slide, everybody, just to sort of round up the day. And thank you to Michael and to all our presenters today, which is Kate Helfer from ALG, who gave us a rundown on the clinical trials through the ALG group, Deborah Sims and her story and her emotional impact of being diagnosed with lymphoma, Clem Bayard from the CNS from Victoria Cancer Council, who gave us some great tips and words of advice of how the emotional impact of cancer. And of course, Michael, with the last presentation and everything we need to know about CAR T cell therapy. I've just shared this slide, so basically just to remind people that, you know, there's a lot of information that's changing quickly, and one of the ways you can keep in touch is keeping up with our resources. We try to keep them as up to date as possible. There is a list on there to show that what the most up-to-date treatments are for each of the subtypes. You can see that on the fact sheet. And there'll be soon a CAR T cell therapy one as well, which we're just waiting on the final draft to be reviewed, and we'll have that out shortly as well. And also to have a look at our videos, the Lymphoma Australia videos. There we've got lots of interviews and presentations. There's one there, a picture there of Michael doing one on diffuse large B-cell lymphoma and overview. He does another great one on relapsed overview as well. Plenty of experts from around the world keeping you up to date with the latest on various subtypes, especially from the recent conference meetings and things. We'll try to keep those as regular as possible. We'll be doing a lot more of these webinars over the coming months, and now we're getting a bit more pro at it all. We only had a few hiccups today, so it's been great. But ultimately, we'd love to thank you all for your support and for joining us today. It's been a fantastic afternoon. So thank you so much, and we'll speak to you all soon. See ya.